Hello, welcome to Biotanemy.
Eukaryotic transcription. The transcription in eukaryotes is more complicated and requires the help of more enzymes to complete. The core promoter element seen in 10 to 15% of the eukaryotes is tau boxes, the same as seen in the prokaryotes. In eukaryotes, there are three types of RNA polymerase. They are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 is usually found in the nucleolus. RNA polymerase 1 will synthesize all RNA except 5S RNA. RNA polymerase 2 is also located in the nucleolus region of nucleus and will synthesize mRNA and several types of regulatory RNAs like microRNA and long-coding RNA. RNA polymerase 3 can also be seen in the nucleolus region and will code for 5S pre-rRNA and transfer pre-RNA. To begin the initiation of transcription, it requires 5 general transcription factors or GTFs. The first one is transcription factor 2D or TF2D. TF2D comprises several tau binding proteins or TAFs and tau box binding factor or TBP. The TBP will recognize and bind to the promoter sequence, or tau box. The TBP will then bind to transcription factor 2B or TBP along with TF2A to form the TBP-TF2B complex at the promoter. The TF2B will serve as a bridge for RNA polymerase to bind to the DNA. The TBP-TF2B complex will now bind to the transcription factor 2F or TF2F. After the RNA polymerase 2 binds to the promoter, 2 additional factors TF2E and TF2H will bind to the complex. TF2H is a multi-subunit factor, which plays the role of helicases, and as a protein kinase, which will phosphorylate the sequence repeats in the C-terminal domain present in the largest subunit of RNA polymerase 2. The phosphorylation of these sequences will release the polymerase from the initiation complex and allow it to move forward along the template, which will start the elongation phase. In the elongation phase, the main phosphorylation occurs on the C-terminal domain, or C-terminal tail of the RNA polymerase 2 molecule. The RNA polymerase will move along the DNA template to synthesize RNA in a 5' to 3' direction by catalyzing nucleophilic attack by 3-OH of the growing RNA chain on the alpha phosphorus atom on an incoming ribonucleoside 5-triphosphates to form a primary transcript. RNA polymerase 2 can continue to transcribe the RNA sequence for a few base pairs more than the actual end of the gene, and the newly synthesized RNA transcript will be cleaved at the internal site before the RNA polymerase finishes its transcription. This site of cleavage is known as the end of the gene. This end usually occurs between AAU-AAA and GU-rich sequence that is separated by 40 to 60 nucleotides in the newly synthesized RNA. The upstream sequence AAU-AAA will be bound by the protein cleavage and polyadenylation factor, and the downstream GU-rich sequence is bound by the enzyme cleavage stimulation factor, forming a protein complex that cleaves the pre-mRNA formed at the sites of 10 to 30 nucleotide downstream from AAU-AAA. The enzyme poly-A polymerase will catalyze the addition of a 3-poly-A tail. The remaining transcript is digested by a 5-exonuclease enzyme, while the RNA polymerase 2 is still transcribing the RNA. For example, XRN2 in humans.
Thank you. Please like, subscribe and share. See you again in a new topic.